Years of scientific discoveries have presented an abundance of information obtained from genomic, proteomic, and glycomic research. This research has provided an insight into the intricate genetic and epigenetic mechanisms. Tools such as microRNA, small interfering RNA, oligodeoxynucleotide transcription factor decoys, plasmid DNA, aptamers, genetic vaccines, and several other polynucleotide forms are helping to transform the methods of regulating gene expression, understanding disease pathways, and undertaking disease therapy. Nucleic acids are most commonly used for disease therapy. This is due to the fact that nucleic acids have a unique affinity and specificity for intracellular targets. For nucleic acids to work effectively, they require delivery systems. These systems compact nucleic acids into nanostructures called polyplexes, which enter the cells to protect nucleic acids from enzymatic damage and further promote target delivery to specific tissues and sites within a cell. All nucleic acid-based molecules are based on either DNA or RNA and their synthetic analogues, such as oligonucleotides. Each of these biopolymers have a high molecular weight and most are highly charged. This can severely restrict their ability to move freely across cellular membranes to reach their active sites within target cells. In 1965, Verheri and Pagano described the first method of nucleic acid delivery. This was the nucleic acid transfection technique. It stated that with DEAE dextran-mediated cell transfection, positively charged DEAE dextran binds to the negatively charged phosphate groups of DNA, forming aggregates. It is these complexes that, when applied to cells, can bind to the negatively charged plasma membrane. It was further suggested that cellular uptake was mediated by endocytosis and assisted by osmotic shock. In 1973, Graham and Van Ebb presented calcium phosphate-mediated transfection, now the most common method of cell transfection. This method is based on the formations of small and insoluble calcium phosphate DNA precipitates. In this process, calcium ions must first bind to negatively charged phosphate groups of DNA. Once phosphate is added to the mixture, it bonds ionically to calcium ions, forming a crystalline complex, which will precipitate. These precipitates' shape and size will affect the overall efficiency of transfection. Later, in 1987, Felgner and others described lipid-mediated transfection. This is a method that heavily relies on cationic lipids to form smaller liposomes with positively charged surfaces. Lipids are commonly used due to the fact that liposomes have a distinct attraction to the phosphate groups of DNA and a cell's negatively charged plasma membrane. When it comes to the use of lipid-mediated transfection, these are some points to consider. It has high transfection efficiency, lower toxicity compared to other transfection methods. It is useful on a larger scale of cells and tissues. It can be used with adherent or suspension cultures, and it is simply easy to use. When it comes to disease therapy, the first and most basic approach is to increase the expression of a target gene by integrating it into viral vectors or bacterial plasmids to further promote expression in tissues or cells to restore the correct pathological gene expression. In recent times, disease therapy has been associated with a wider variety of strategies using short oligonucleotides. This is a unique antisense-based method used for its high specificity to target virtually any single group of genes. More precisely, these methods can include classical antisense oligonucleotides, splice-switching oligonucleotides, anti-gene oligonucleotides, small interfering RNA, microRNA, and anti-microRNA. Single-stranded antisense oligonucleotides act primarily in the nucleus by selectively splitting pre-mRNA that have complementary sites via ribonuclease H-dependent mechanisms. Currently, these antisense oligonucleotides are used as gapmers with a central region that supports RNase H activity surrounded by chemically modified ends that will increase affinity and reduce susceptibility to nucleases. Splice-switching oligonucleotides are typically modified to ablate RNA's H activity and allow interaction with pre-mRNA during the splicing process. They can bind to either the 5' or 3' splice junctions or to exonic splicing enhancers or silencer sites. By doing this, it may promote an alternate use of exons, exon exclusion, or exon inclusion. In small interfering RNA, the selective splitting of mRNA in the cytosol involves Argonaute 2 containing complexes and requires mostly complete complementary siRNA template strands and the target strand. In microRNA, complementary 3' untranslated regions lead to translation, followed closely by message degradation. A single microRNA can easily influence the expression of multiple mRNAs. While this has the potential to be a problem, it can also provide coordinate regulation of an entire set of genes. Antisense oligonucleotides can also act as antagonists to microRNA, potentially increasing the expression of microRNA-regulated genes. Nucleic acids are most commonly introduced into cells via transfection, a process whereby nucleic acid sequences, as well as the proteins and oligonucleotides, are presented by either a biochemical or physical process. Some examples of biochemical approaches include the DEAE dextran, calcium phosphate, liposome-mediated transfection methods, and nanoparticle delivery. Physical transfection methods, however, may include the direct microinjection of materials, biolistic particle delivery, and electroporation. Lipid nanoparticle delivery is the most commonly used and clinically advanced approach, compounding anionic oligonucleotides with cationic lipids. This approach has been most important for the therapeutic use of small interfering RNA. Lipid nanoparticles that are used in vivo are typically 100 to 200 nanometers in size and have a surface coating of a neutral polymer, such as polyethylene glycol. This minimizes protein binding and uptake by RES cells. By doing this, there is a greater persistence in circulation and opportunity to interact with other cells. Though, due to their small size, lipid nanoparticles can only exit circulation sites where the endothelial barrier is perforated, such as in the liver, spleen, and certain tumors. Because of this, most siRNA and lipid nanoparticle disease therapy is focused on liver diseases, clotting disorders, liver cancer, and disorders of the lipid metabolism. Lipid nanoparticle delivery involves initial uptake by endocytosis. Once inside the endosomes, cationic lipids interact with anionic membrane lipids to disrupt the membrane structure via the formation of a non-bilayer lipid phase. This leads to increases in membrane permeability and the dissolution of lipid nanoparticles. It is the basis for transporting the oligonucleotide to the cytosol. Polymeric nanoparticles provide another strategy for nucleic acid delivery. It is worth stating that the first use of small interfering RNA to treat human cancer involved a polymeric nanoparticle. Typically, these nanoparticles are formed by the self-assembly of amphiphilic polymers in a water environment. Commonly, a triblock polymer may be used, such as a hydrophobic portion to drive self-assembly, a cationic portion to bind the oligonucleotide, and polyethylene glycol to provide a protective coating. This system works most effectively in a long circulation lifetime and has shown proven effective siRNA delivery to tumor xenografts in mice. The delivery of nucleic acids can be divided into two main applications: viral and non-viral delivery. In most cases, viral vectors are remarkably more effective in delivering genetic materials into cells. Due to the recent developments in vector design and safety, viral gene therapy applications have made revolutionary steps for several genetic disorders in disease therapy. In addition to this, several gene therapeutic platforms have been discovered. These include gene editing platforms such as zinc finger nucleases or transcription activator like effector nucleases. These are used to access and alter the genomic DNA. These platforms can also chemically stabilize mRNA that could be used as templates for protein synthesis and/or modified mRNA. It has also been suggested that these molecules have allowed to significantly expand the space of pharmacologic target, otherwise undruggable with conventional drugs. One of the first major antisense drugs approved in 2013 was called Carnimro, an inhibitor of apolipoprotein B expression. It was accompanied by clinical trials involving small interfering RNA and splice-switching oligonucleotides. Over the last few years, clinical trials applying a wider variety of oligonucleotides reported groundbreaking results. Some examples include receptor-targeted siRNA conjugate, strong effects on liver disease using antisense with unique chemical modifications, anti-cancer effects using miRNA, and the treatment of neurodegenerative diseases via splice-switching oligonucleotides. Recent applications in disease therapy have heavily relied on the progress in the medicinal chemistry of nucleic acids. One such progress is the modification of the phosphorothioate backbone. This backbone creates a modest reduction in binding affinity. Its two most noteworthy advantages is to improve stability to nucleases in blood and tissues and to promote protein binding, thus supporting interactions with albumin and other blood proteins.
The delivery of nucleic acids for disease therapy has enormous potential for the prevention and treatment of many diseases, including cancer throughout the body, liver disease, neurodegenerative disease, gastrointestinal disease, and dermatological disease, just to name a few. The advantages of the delivery of nucleic acids for disease therapy is many, but to name a few, patients will have an increase in treatment possibilities for genetic and inflammatory diseases. In some cases, where there is currently no treatment, such as for Machado-Joseph disease, there is potential for treatment options to become available. Treatment options for patients are currently limited and can be invasive, such as surgery, or can require repeated injection or infusion. It is hoped that the increased treatment options and potential cures will lead to positive outcomes for patients through reduced physical and emotional pain of chronic disease. Although enormous potential surrounds the technique, many articles report on the challenges surrounding the use of nucleic acids for disease therapy. The shortcomings of the technique lie in the delivery itself. To use nucleic acids in vivo for disease therapy, the nucleic acid must reach the cytosol or nucleus of the cell within the target tissue. For this, there are two main issues. Firstly, the nucleic acid delivery must reach the target tissue, whilst exposure to non-target tissue is as little as possible. Secondly, the nucleic acid must be transported to the intracellular compartment where it is required. Some examples of the shortcomings of this technique are as follows. The kidneys are an important factor in pharmacokinetics and biodistribution, especially in the concern of excretion of siRNA. Renal excretion can occur rapidly of siRNA and uncharged oligonucleotides due to their low affinity in binding to plasma proteins. The siRNA are mostly less than 3 to 6 nanometers in size. Molecules this small are ultrafiltered and cleared rapidly by the kidneys. In the instance of antisense oligonucleotides, the ability to bind to plasma protein is high. Therefore, renal excretion of antisense oligonucleotides is reduced and allows them to be further distributed throughout the body. In both of these scenarios, antisense oligonucleotides and siRNA accumulate mostly within the abdomen area, especially the liver and kidneys, which influences the distribution of molecules. To successfully reach target tissue, as we have just seen, systemic administration may be unsuitable. Filtration and excretion, unuseful accumulation in healthy tissues, and degradation by enzymes can all interfere as the nucleic acid travels within the circulation. It is hoped that oral administration of siRNA can be used in the treatment of gastrointestinal diseases due to the increased bioavailability local delivery permits. Although, as the gastrointestinal tract is designed to prevent foreign molecules from entering the body, the challenges surrounding oral delivery are many. siRNA, when administered orally, faces many obstacles, such as low stomach pH, which can cause a depurination chemical reaction, enzymes within the gut lumen, which can cause degradation, intracellular and extracellular barriers, making it physically difficult for nucleic acid to reach their target successfully. To reach its target, physical barriers such as mucus layers and epithelial cell layers must be breached. Phagocytosis must be avoided so the body does not remove the foreign molecules. And as both the siRNA and the target cell surface are negatively charged, they repel one another. Side effects resulting from the delivery of nucleic acid for disease therapy include oncogenic potential, the potential to result in cancer, genetic damage, and immune response is seen in patients administered viral vectors, even though rates of transfection are more promising using this method. Interaction with pattern recognition receptors can cause inflammatory responses via the innate immune system in the delivery of ASOs and siRNAs. Potential issues may arise from the delivery of oligonucleotides through the interference with blood components and intracellular proteins. Looking further into the potential of nucleic acid delivery for disease therapy, many strategies are currently being investigated. Some examples are chemically altering the oligonucleotide, for example, phosphorothioate backbone modification, which improves stability of the molecules within the blood and tissue and promotes protein binding, therefore reducing renal clearance. This technique currently comes at the cost of reduced binding, affinity, and toxicity. Using agents that target receptors, for example, carbohydrates or peptides, which can be linked to the oligonucleotides to assist transportation. Using nanocarriers to transport the oligonucleotides. There are currently several types of nanocarriers, with polymers being one of the most researched. Very little knowledge currently surrounds polymers as nanocarriers on a cellular level, but research shows that biodistribution of oligonucleotides may be substantially improved with further development of this technique. Thank you for taking the time to view this presentation on nucleic acid delivery for disease therapy, which was conducted by Cara and Megan as part of the BCM201 Fuller Trobe University.